only not just to do the natural thing, which is answering peerwise questions, because great, kind of like a question bank, but really to try to ask questions and to ask difficult, higher order level questions. The more you do that, the more you're allowing your brain to practice that mindset of the what-if scenarios, right? You can memorize as much as you want and go over the notes. All of these are very important, but again, you know me, and I'm not going to ask you too many questions in the exam about is the SA node in the right atrium or left atrium, right? I mean, this is not my job. So we talked about the conductive system. Today I'm going to ask you about what happens if question, but there's so many what happens if, so ask yourself. So just a reminder about P1s. So last time we discussed intensively the conductive system of the heart. Today we'll just finish it, talking mainly about the homeostatic imbalances leading or related to a conductive system. And we'll talk about the second part of the heart, which is the contractile. So we'll talk today about the cardiac cycle and how the firing of action potentials lead eventually to contraction, how the blood flows in the heart, and we'll just do it one step at a time. We'll talk about the valves, how they respond in different steps of the cycle. And the last thing that we will do today is talk about some physiological determinants, basically volumes and pressures, and that will be the beginning of our discussion that will continue next week on how we regulate cardiac function. Okay? So that's what we're going to do today. So I want to summarize. Last time we went step by step, just a short animation to show us everything, and I'll do it. We'll see. We start with the SA node, right? The action potential fires to all the blue labeled cells. I'm just showing the right side, but the same thing goes for the left side. Here we had just a delay that is essential because we don't want the ventricles to contract at the same time as the atria contract. Now you see here a delay. This delay doesn't exist. Okay, the delay is just for didactic purposes for the animation. But here the action potential will split to the bundles of His. Not waiting here, this is just because of the animation, and then going up. And now if we add contraction, then what we will see is action potential contraction. This is still waiting and then contracting, and again contracting from bottom to top. We talked about the idea of pushing the blood from the apex, from the bottom, all the way to the top, to the pulmonary trunk or the aorta, depending on the specific ventricle. So, again, as always, one of the best ways to learn about a function is to ask what happens if this function is compromised. We have many, many alterations, abnormality when it comes to our conductive system, and we put them under one name that is called arrhythmias. Okay, basically something is wrong with the rhythm. You cannot say it even more simply than that. Defects in the intrinsic conductive system, for example, the SA stops functioning, or the AV stops functioning, or some cells stop functioning, or everything is functioning but there's no hierarchy. Some cells that should have responded to the other cells, to the previous cells, are starting to pace by themselves. Now we're not going to cover each and every arrhythmias. You guys will go to medical school at some point, especially to be cardiologists, will know each and every detail about this, but we're going to talk about some concepts, and I do want to mention one of the amazing, basically, developments of modern medicine is the artificial pacemaker. And the idea that coming back to the fact that as much as this machine we call a heart is complicated, it's basically a machine, and that allows us to, okay, we have a problem with the machine, but we can replace the machine. And that's basically what we do. So if we have a problem in one of the components of the conductive system, we can actually replace that. And what we do, we put a pacemaker just beneath the skin with electrodes. It used to be very simple, not simple, it's very complex, but it's relatively simple to what we have today. And to put it basically on the SA node and to replace the SA node, today we can do so much more than that. We can put the electrodes in different places. We can actually make it dynamic. It used to be very static, but to make it dynamic so it will respond, we'll have sensors. So if you run and you need the heartbeat to increase, we can do that. You can today have your doctor check your activity not in your presence, but basically through different monitors and through the phone, basically change the set or reset the pacemaker. It's pretty amazing what we can do today. But the basic idea, the basic concept is because we are talking about very basic electrical stimulus. The action potential, right, is basically electrical stimulus. What we can do is just provide this electrical stimulus instead of the SA node cells or AV cells or whichever cells those are. That's the idea of the pacemaker. So let's try to think about the cycle by trying to answer this question. Okay, so read it. Start talking with your friends, and I'm going to open it. What happens if scenario? Now, I want to hear those who chose B, what was the rationale? Why would you think B is the correct one? Dina, exactly, and that's the part that maybe was missing for many. It's the second fastest node, the cluster of autorhythmic cells that reach a threshold. So do remember, all the pacemakers have that potential. I would argue probably if I had to answer this question and I didn't know the fact that the AV is the second one, I would be tempted to do C. Their issue is that, well, yeah, maybe it's not exactly like conflicting with A, so I can understand C actually. So, but the issue is now is that basically AV is the second fastest. So if we go back to that parade, right, and the drum major leading and being the fastest and everyone is following, the drum major is sick or unfortunately got a heart attack, no pun intended, during the parade. But the second fastest, right, leader will become the drum major, and that person, AV node in our case, can just leave. Having said that, yes, here. Okay, so I'm actually, Sierra is asking a great question. What about, I mean, we have the, and that is going to the left from the right, right? From the right to the left, right? So we have a problem with the right atrium. But even I'm going to actually expand on your question and think for a second. So you have the AV node, right? And okay, we can have the AV node and we'll just kind of like cut it here and we'll continue and then we'll have contraction of this ventricle and contraction of this ventricle, right? Because the left ventricle, remember, is independent of the left atrium. The conductive system is working from the SA node and then AV node from the right atrium. So we are okay with the left and right ventricles. Will they contract at the same pace as they're used to? No, because the AV is slower. So instead of, let's say, about 60 beats per minute, maybe it will be 40 or 35. Now, this is not a good thing. You will want, if someone has this arrhythmia, you will want to provide this person with a, yes, exactly. So you're going to make, back right with the artificial pacemaker just to replace this. Because that person wants to run up the stairs and you need sometimes to increase the heartbeat, that's true. But at the same time, it's not that someone is just going to die because you don't have an SA node. Your heart is still going to beat, just slower. But my question, expanding what Sarah was asking, is how come? Because we just talked about the ventricles. Can the atria contract if I don't have an AV node? Left and right. They cannot. How come? Where are they going to contract from? Right? I mean, we don't have this going up. We started here, we take care of the ventricle, but this is a dead end. So the atria are not going to contract. So why is it a problem? Potentially at least. Question first, okay.
When you said that it's going to be slower, does that just mean like a lower heart rate?
Yes, it's going to be slower, it's going to be a lower heart rate, exactly. So, why do I care if the atria contracts or not? The problem here is with the SA. We start everything from the AV instead of the SA. Okay, the SA was compromised for whatever reason, temporary, not temporary. AV is taking over. We are good. Ventricles can contract. What do we need the atria to contract for? What is the job of the atria in general? To push blood to the ventricles, right? So the ventricles can contract, that's wonderful, but the ventricles need to get blood. And they're not connected to the vena cava, right, or to the lungs. Basically, they need to get the blood from the atria. So we need to remember this problem, because it sounds like that person cannot live for a second, but that person can. Okay? How? Later today. Just remember this question. Okay. There are, again, as I said, multiple arrhythmias, but I just want to address one more, which is kind of like the worst arrhythmia, that basically will kill you in a very short time if it happens, and that's what we call ventricular fibrillation. Ventricular fibrillation is when not that the pacemakers or the autorhythmic cells stop firing, but they stop following the hierarchy. Each and every one is pacing at its own pace. And this makes, if you don't have a hierarchy, our parade goes everywhere and anywhere, right? And you will not be able to contract the atria first and the ventricle next, and everything is going just not efficiently. That will lead to death. And this is where I think Carl was asking me last time, right, about the pedals. This is when we do what we call defibrillation. You see probably in the movies, and hopefully none of you encountered that. Anyone encountered that? Saw that like in real life? Action of that? So what's the idea of defibrillation? Basically, the system is a short, if you will, right? Everything is just going crazy. All the autorhythmic cells are fine, but they're not organized. In defibrillation, what we do, we make a short. We cut the whole system. We break the system. And what we hope, and sometimes it helps, sometimes it doesn't, that when the system is going to restart, it's kind of like reload your computer when your computer goes awry. Then again, the SA node, if it's functioning, will take over and will lead and will go back. Yes?
Does ventricular fibrillation correspond with a heart attack?
Not necessarily. So the question was whether it corresponds with a heart attack. Not necessarily. Okay? Yes, here.
Why would it take sometimes multiple shocks? Like, why would it be different? Like, I've never seen this in real life, but in movies, sometimes the first one.
Have you ever had the opportunity to indulge yourself with a computer crash?
A what?
Did your computer crashed? I mean, you had to reload it or something like that, but the first one did not work, and the second and the third, that's exactly the same. Basically, sometimes it works, sometimes it doesn't, and sometimes that shock happens in the third time. Of course, it will not always work, because sometimes those systems just don't function. But it does save a life. That's the idea. Again, at basic, it's all the conductive system. It's all basically electrical impulses. It's the turning on the switch, going to the lights. The same idea. Okay. And I put this this link, just if you want to follow it, you can learn a lot about different arrhythmias. And you have a Sakai assignment. I'm just mentioning it because I think it's the first time. Yes, you have a Sakai assignment that is not a G or Q assignment. So you have a Sakai assignment about arrhythmias. It's basically to describe some arrhythmias and just read the details here. And I'm just reminding you that it's October 29th. Okay, post-class assignment. Before leaving the conductive system and starting to talk about the cardiac cycle and how we contract and the blood is flowing, physiology is unlike, I'm teaching genetics. Some of you are my students in genetics, and someone who knows anything about molecular biology, this is a field that what you learned 2 weeks ago might be very different now. Okay, it's so rapid. Physiology is not like that. Okay, there's a lot of things that we know that I teach you that we have known for 70 years and sometimes even more. But it is an ongoing field. There's a lot of research. Just one example of an interesting research is something that I wanted to show you here. So we talked about the fact that if you want to make one cell beat and that the next cell will beat afterwards and be all coordinated at the same pace and everything, we need to transport, to propagate the action potential, and we do it through what? Gap junctions, right? So at least theoretically, if I take two heart cells in the lab, and separate them physically, distant enough that they don't have any gap junctions, and I let one beat, What will happen to the next one? Will it beat? No, because there are no gap junctions, right? They're not going to be coupled. But here's some interesting research. So this came from a lab in the Technion, my, not exactly alma mater, but when I did my PhD in Israel, and mechanical communication in cardiac cells. Okay, it's from 2009, and Idonit Fan and Stavit Rory are co-first author on this. The reason I mentioned this is Stavit. I'm just showing off because she was my lab mate for many years. She was a grad student. And now she's a research assistant professor, and she was one of the two that led this study. And what they did is to take two heart cells, you're familiar with that from the Facebook from last night, and they put them on a table, but not just a table. It was a very specific table that can basically sense and rotate, or not rotate, but I guess responds to any sort of physical touch. So if you push on the desk, then another part of the desk will also be pushed. State of the art, modern, it's biomedical engineering department. Okay? And what they saw is that if you take cells, this is actually the response. You take one cell that is what we call dormant, okay? Just a contractile muscle cell, but it's not something that right now beats. And you put another one and that one beats, and the other one, so the first one, start beating, but not only start beating, but exactly at the same pace. They're synchronized. Now, there are no gap junctions. They are way, way far from each other. Can we explain it in maybe another way that is not mechanical? What else can we hypothesize is working here? If they're still pretty close to each other.
Mechanical stimulus?
But if it's not mechanical stimulus, can you explain it any other way? Think about some, yeah. Maybe it's chemical signaling, right? Maybe the first one, I don't remember which one is the first one. Let's say the first beating cell is the right one. And maybe it secretes some molecules, right? And as a result, that will lead to. So they did something really clever. They said instead of like having two cells, they take one cell and a probe, a mechanical probe that will beat on that same table. Okay? So they have the cardiac cell here in red, and they have the fancy table or platform here. And now what they have is some mechanical thing, a mechanical thing that does not secrete any chemicals. Still, they beat. What it's showing us is that actually just the mechanical contraction of one cell can potentially lead to contraction of the other cell. Now, this doesn't necessarily mean that this is the way it works in the heart. It does mean that those cells have the potential to do that. Whether this applies to real life, whether this is kind of like a backup mechanism, we don't know yet. Not at this point of the research, but just to show you that we continue to learn new surprising findings about physiology, and in this case about conductivity. Okay? And I showed you already that, but I showed you when we talk about the skeletal muscle, this was actually a heart muscle, and this is from their study. Okay, that's those cells contracting. Each of them, we didn't talk too much about this, but the heart cells at the molecular level, the same thing as we learned in the skeletal muscle cells, tropomyosin and everything that we discussed. Okay. Because this is how they design the table itself. So it's not really something that you just jump up because the table is moving. It was sensitive enough, and again, don't forget, we're talking also about synchronized beating. So it's beyond that. This is a real cell that contracts, and you can look at the, again, fibers and see everything is already working. Yes, Maliki. No chemicals and no gap junctions, so that means it has to be mechanical simulation. Okay? There is a mechanical, one beats one way or the other. The next one is just filling. I'm using the word filling. Of course, it's not really filling. But getting the beat, the mechanical as a stimulus, and that leads to beating. And again, I emphasize, we don't know whether this really happens in real life, in vivo. We just know about the potential. If you take the stimulus away, the cell will not, if it started to beat, it can continue to beat. I don't know for how long, but it can continue to beat, yes. Okay.
Is that the idea behind like a cardio?
a cardiac massage? Basically, I don't have a good answer for that. I just don't have a good answer for that. Okay, we talked about the conductive system. Let's see what we get from the conductive system. We know how to fire action potentials. Let's talk about the cardiac cycle. So what is that cardiac cycle? Cardiac cycle involves a lot of things, and I will not ask you to learn all these things. We are not going to talk too much about volumes. We will not talk about heart sounds. Many interesting things, but for the level of this class, we'll focus on some important factors. But basically, the cardiac cycle refers to all the events associated with blood flow through the heart during a single heartbeat. Not a minute, not half a minute, a single heartbeat. The main thing to remember is something that you basically know. In order for blood to move from point A to point B, we need to have a gradient. Just like in order for sodium ions to move from point A to point B, we need to have a chemical gradient. When we will learn about the respiratory system, we will see that in order for air to flow from one point A to point B, we need to have a pressure gradient. In the case of the blood flow, that means we need to have differences in pressure. First of all, I want to make sure we understand the term pressure. Pressure is force per area. So that's the force per area that the blood molecules, the blood fluids exert on the walls of the chamber. So if you are sitting in the wall of the atrium, let's say the right atrium, and you're hit by the blood, if you have blood inside, you are getting pressure, force per unit of area. So in order for us to make blood flow because we want the blood to flow, right? What we are doing in the heart, we are creating that pressure change. How do we create that pressure change? By contracting the muscles. That's the idea. This is why we need such a strong muscle in the heart. And this is what we're going to discuss. You're pretty familiar, I'm sure, about the term systole and diastole. You heard that, but now it's time to learn that the ventricles have their own systole and diastole, and the atria have their own systole and diastole. We know that they're not acting in unison, right? Systole, contraction of the heart muscle. Diastole, relaxation of the heart muscle. And again, both atria and ventricles have their own systole and diastole. If you don't hear, if someone is talking, if you go and measure your blood pressure or something like that, or if you talk to the doctor and they say something about systole and diastole, they typically don't say ventricular or atrial, right? So if they don't mention anything, what we typically talk about, the default is the ventricle. Why the ventricle? Because the ventricle are much heavy muscles because they need to push the blood all the way to the lungs or to the entire tissues, while the atria just need to push the blood to the neighboring ventricles. Okay? So the default, if we don't say something specific, is that it's ventricles, but we, of course, will talk separately about ventricles and the atria. Okay, so we're going to start with the cardiac cycle. Unfortunately, we don't have the time to talk about ECG or EKG. But I'm going to use the EKG or ECG graph as a background, so I want to explain in a word about this. EKG or ECG, because we have so much electrical activity in the heart that we already discussed, we can actually measure that activity even at the level of the skin. It kind of looks like an action potential, but it's not an action potential. It's the sum of all the activities that you find there. This is a typical ECG of a healthy young person. And I'm just going to mention some highlights again. And I'm going to ask you questions in the exam about what is this and what is that. It's just to understand what we have in the background. Because we need to link the conductive system to the cardiac cycle. P is the first firing of an action potential in the cycle. Where do we start our conductive system? Guys, where do we start it? 1, 2, 3.
P.
SA node, wonderful. So P is that activity, okay, or the depolarization, if you will, of the SA node. QRS is the depolarizations of the autorhythmic cells in the ventricles. Now, it's much more complex than the P, and there's a reason for it because there's so many things going on, and remember that autorhythmic cells, it goes down and then up and so on. We just call it QRS wave. And finally, we have the T activity, which is a repolarization of the ventricles. Again, I'm not talking about contraction, relaxation of muscles. This is nothing to do with muscles. This is just electrical activity. This is just the conductive system. Why don't I see repolarization of the atria? Because it's masked right here. It's in that QRS. You cannot just see it separately. Okay? So again, I want to emphasize I'm just using it as a background, as a wallpaper, if you will, because we're going to link it to the contractive system. Now, it's a cycle. We can start a cycle anywhere. I'm going to decide by starting here at the end of the previous cycle. So we'll define a start and an end. This is random, of course. We will start just before atrial depolarization, the SA node, and of course the actual contraction. Okay? And what we're going to do, we're going to talk about one step at a time. At each given step, we will talk about three things, or three factors. We will talk about the atria and the ventricles, are they in systole or diastole? We will talk about the status of the valves, okay? The AV valves and the semilunar valves. And then, of course, we will talk about the blood flow. And as you will see, systole, diastole in black font, valves in green, and the blood flow in red. Just to make sure you are following this, okay? So before we even start, if this is the end of the previous cycle, let's talk about systole and diastole. What do you think the ventricles and the atria are at this point? They're in diastole, right? Remember, contraction is systole, relaxation is diastole. So we are after relaxation of the previous cycle. So let's see what we have here. So here is a diagram of the heart. Every now and then I will use either the left or the right, but they all apply to both, okay? So we are in late atrial and ventricular diastole. All the chambers are now relaxed. The AV valves, the valve between the atria and the ventricles, are open. Why exactly? This is due to the previous cycle. We'll come back to this at the end of our coming cycle. Right now, just take it as a given. And we have some ventricular filling. Again, why is that? Because of the big previous cycle. So we just, right now, just think about it as this is our starting point. Now let's start. We're reaching the P. P is firing, right? It's the firing of the SA node. When we ask about contraction, which part of the heart is going to get into systole? I'm going to think about it for a second, and then I'm going to count to 3, and you're going to tell me. I'm asking about systole now. 1, 2, 3. Atria, wonderful, right? It's going to be a tiny bit after the P, because first of all, we need the action potential, and then we're going to have contraction. What about the ventricles? Are they going to go immediately to systole? No, right? Because we talked about that very important delay. Okay, so let's see what we have. In each step, we will have the previous step diagram and then the next step diagram, okay? So we're talking about number 2. So the atria are getting into the systole, and you can see the systole by this kind of like change in the angle, something like that, right? Right here in the skeletal wall, in the, sorry, not skeletal, in the atrial wall. So they physically tract. The ventricles are still relaxed because of the delay in the AV node. AV valves are still open, semilunar valves are still closed, nothing changed here. And we have ventricular filling from the atria to the ventricles. That's their job, right? Contracted, remember that plastic bag with water that we discussed last time? We contracted the walls of the atria. Blood is going from higher pressure to lower pressure. By contracting, we increase the pressure, okay? Remember, pressure is force per area, so I just increase the pressure, the force. Sorry. And now the blood is ejecting towards the ventricles. Any questions about step number 2? Yes, Nicole? I increase the pressure in the atria by contraction. So now I have greater pressure in the atria than I have in the ventricles. Blood flows from greater pressure to lower pressure. Remember, the ventricles are relaxed at this point. Any other question? Okay, The next step, again, you guys will help me even before we look at the step. Don't look at the next slide because it will help you to think about this. So now we had our systole in the atria. What's going to be the next step with the atria first? After we contracted, we are going to go into diastole. We're going to relax. What about the ventricles? They were very polite. They waited nicely, right, until they get all the blood. But they're going to go now into systole, right? That's the QRS electrical activity that will lead to actual physical contraction. So now we're moving from 2, that was the previous step, to 3. Ventricular systole, the ventricles are now contracting. I know it's really hard to see that in the diagram, so just imagine they are physically contracting, pushing the blood. The atria are getting into the diastole. And now let's see what goes on with the valves, because the valves are now changing their status. So take a look first of all on the AV valves. Okay, AV valves were open. Of course they were open. The blood was being pushed and opening them. Okay. But now when the ventricles contract and the atria relax, they're closed. How come? What's going on here? You guys read about the valves. What's going on here? What made them close? What are the muscles that push them to close? That was a kind of like a similar question, not exactly in the GRQ. What pushed them closed? What made them close? It was the blood that pushed them to close, right? The valves are kind of like the Wild West movies at the bar. You had that door, the right, that can open both ways. So we had blood pushing through the door from I'm in the atria now, okay? And let's say I'm pushing now the door open. I'm the blood molecules that move. I'm now in the ventricles. So now when the ventricles contract, I'm pushing the door back towards the atria. So why aren't they opening towards the atria and the blood goes all the way from the ventricles back into the atria? Because not exactly the muscles. What else? What else do we have there? These are the valves themselves. They are one-way valves. So what are those muscles and cordia? What are they for? They are there to make sure that when I close that door, I close it up to a point. Okay? And I'm not pushing it all the way towards the atria. The blood will not flow. But what we do, we push them, but at the same time, they are the cordia, with the contraction of the muscles, basically saying they're pulling, so they're closing, but up to a limit. And then we hold them close. They're not bulging into the atria. So these are the AV valves. But here's something really strange. The ventricles are contracting, right? They're creating pressure. I even put the letter P for pressure. Okay, let's talk about the left side. Again, the same thing goes for the right side. The semilunar valves are still closed. I'm pushing those doors. The direction of those is toward the aorta or the pulmonary trunk, depending on which side. I want you guys to think together and tell me, why aren't those doors opening so the blood will flow into the aorta? Is the question clear? Clear? Okay, again, I do create a pressure here in the ventricles. The blood cannot go to the atria because we closed everything. But why don't the semilunar valves open up? I will just give you 2 hints. One of them is that there's something missing in this diagram, but at the same time, there's a letter P that is there that should help you. Try to think and talk with each other. Question is, why don't those semilunar valves open up? Guesses? Anyone wants to suggest anything? Yes, Nicole, give it a shot. Wonderful. I'm stopping here for a second so we can talk about it one step at a time. The P represents pressure. But I put P, big P, right, or large font right here in the aorta. And again, I didn't put the pulmonary trunk, but it's exactly the same story. So there is a pressure built here, but in order for the blood to move from point A to point B, it has to be in a greater pressure than the other one. And right now, in the beginning, if I'm the blood, okay, I will represent the blood. Here's right here. I don't have a door. So can you imagine the valves? Okay, this is the door. The ventricles, the aorta is behind this door. I'm trying to push the door, right? There's a pressure here, and I'm trying to push even more and to push even more. Myosin heads, binding to actin filaments, starting to pull more and more and more. But there's a greater pressure on the other side of the aorta. So let's follow this. What is that pressure? Where is that pressure coming from, though? That's the part that is missing in this diagram. It's not gravity. It's not gravity. What do you mean when you say, be more specific? We didn't learn yet about the resistance, but just in plain words of what are we talking about? I think you got it right. The pressure behind that door is coming from blood behind that door. Okay? So, again, coming back to my I'll be the blood, I'm trying to push that door. The problem is, okay, I'm moving, if you will, for a second, to the aorta right now. There's someone leaning against the door. I'm trying to push the door, but there are maybe 3 people leaning against the door and make it hard. What are those people? They're blood as well, in this analogy. Where's that coming from? What do we have? Why do we have blood there? This is missing, right? So we need to have actually some red blood here. Where is that blood coming from, though? From? This one is not the pulmonary system. This is the aorta. Right? Any ideas? Anyone wants to suggest? Yes, here.
Is it still from like the previous contraction?
Perfect. That's the blood from the previous cycle. So now we can wrap it up. Blood that will go beyond into the aorta will be sent, as we will see shortly, to the tissues, and if it's the pulmonary trunk, to the lungs. But not all the blood is going to be there. Some of the blood is going to stay there, and that will be the one leaning against the door. Like, if you don't build pressure in the aorta, you'll have a problem because you're going to push too much blood and too quickly into the system. That's actually going to be very important. Next week, we'll talk about the vascular part of the cardiovascular system, and we'll see how important that is going to be. So coming back to that pressure. Yes? I'd like to you guys think about what will happen if. Great. If you have air in the circuit, that's going to be a big problem. Okay? Again, you need to build pressure. The pressure is built by blood. You're going to mess it up. Let's see. So right now, again, internal pressure is generated in the ventricles. Let's do a short learning catalytics. About isometric, isotonic, we talked about movement and pulling bones. Here we're not pulling bones, here we're moving blood from one point to the next. So try to apply this to this scenario. And we'll do it quickly. If everyone, but the majority of the class chose true, and that's true. That's basically the same terminology, but we apply it because we don't pull bones here.